Assalamu alaikum. Today, inshallah, we'll be continuing your immune system. And we covered during the last session or the last video, we covered the innate immunity. Today, inshallah, we'll be covering adaptive immunity. As a recap, as a quick recap, we know that the immune system is subdivided into 3 lines of defenses. The first line, the second line, and the third line of defense. Okay, so immune system. Based on the line of defense, first, second, and third. The first and second line of defense, we consider them as the non-specific immunity. And why do we call them as a non-specific immunity? Because simply the antigen of the pathogen, which is the surface protein of the pathogen, will be recognized non-specifically. So there, I encounter any pathogen, I encounter any pathogen. But during the third line of defense, no, you have a specific antigen, and this specific antigen will be killed whether by a specific antibody, AB stands for antibody, or it will be killed by specific cytokines. So this is the third line of defense. We'll go back to this picture, but let's first subdivide what third line of defense is. So you have the specific immunity. Another name for specific immunity is the adaptive immunity. Also, another name for adaptive immunity is acquired immunity. All of those will be subcategorized into 2 major forms. One of them is called the humoral immunity. Humoral immunity, while the other one is called cellular immunity. Specific, adaptive, acquired, all of those are the third line of defense. They represent the third line of defense for your immune system. Yet you have to know that your third line of the humoral immunity and cellular immunity have different names. Humoral stands for the tissue or the liquid part of your body, the liquid part. So it will be also called antibody mediated immunity. And we'll talk about this in details later. Cellular immunity has a name called cell mediated immunity. This is like the major branch of what cellular immunity is and what an antibody mediated immunity is. Yet in order to recap, let's start with the last one. We agreed that when we were in tissue fluid, there will be so many drawing over here right now. We are like this, cells, and the cells will have been separated with some tissue fluid like this. Tissue fluid, and those are the cells. Those tissue fluids were supplied by a blood vessel. So this is a blood vessel. The kind of cell that's found within the tissue fluid originally after just being made within the bone marrow are called the mast cells. I know that you managed to remember. Those are the mast cells. When the mast cell starts to release histamine, the material that will be secreted within the tissue fluid to the blood vessel, a process called vasodilation will be taking place. When vasodilation happens, you have 2 kinds of cells that will be removed out of the blood vessel to the tissue fluid. One of them is the small phagocyte called neutrophil and the large phagocyte called macrophage. Both of them will be encountered in the tissue fluid. Now they are no longer within the blood vessel. They are here within the tissue fluid. When both of them will be doing a process called phagocytosis. So once again, what phagocytosis is when the white blood cell uses the digestive enzyme in order to kill the pathogen. Any pathogen, I only encounter a pathogen inside using the digestive enzyme and I kill the pathogen. Yet, this is what really happens for the neutrophils and macrophage. But macrophage, and this is the transitional phase, would you please focus? We call this as a transitional phase of the immune system. Transitional phase. This is very important. This is the phase where you are changing from the second line to the third line of defense. During the second line, you covered phagocytosis. You fully finalized the process of phagocytosis. Like macrophages is a kind of cell that can do two main specific functions. So this is a macrophage. When macrophage is playing within the second line of defense, it will do a process called phagocytosis. Then within the transitional phase, within or during the transitional phase, it will do another two type of process. Number two, it will act like an APC. And number three, it will activate the T helper cell. APC stands for antigen presenting cell. What do I mean by this? Okay, let's have a new blank slide and start to draw. Okay. Now this is the macrophage. This is the macrophage. When the macrophage encountered the pathogen, this is the pathogen. Each pathogen has a surface protein called antigen. This is the name tag, or okay, this the label or the name tag. We can keep it between brackets, called it the name tag. It's an identification for the pathogen. When the pathogen started to encounter it, it will look like this. Now the pathogen encounters, the macrophage encountered the pathogen inside, along with the antigen on its surface. By the digestive enzymes, we can draw the digestive enzymes like this. By the digestive enzymes, it destroys the pathogen. So this stage is part of the second line of defense, which is phagocytosis. And where does it happen? It happens in the tissue fluid. This is very important. Tissue fluid. So now you have the pathogen non-specifically destroyed by the macrophages during the process of phagocytosis as a part of the second line of defense. But in order to start the transitional phase, you have something called APC. What do you mean by APC? After macrophages destroyed it, it will act like an APC. It would look like this. Now, this is a macrophage, yet instead of having the antigen as part of the pathogen, it will take the antigen on its own surface. Now the antigen of the pathogen is represented on the surface of macrophage. This is the antigen of the pathogen. And this is the macrophage surface. Macrophage surface. And APC, as we said, it's called antigen presenting cell, because this is the cell presenting the antigen on its own surface. Why do we need this? Because we need this for the specific immune system. The specific immune system, in order to start working specifically, it must get acquainted or be in the know of what the antigen I'm fighting, what the type of antigen I'm fighting. So the macrophage present the antigen for both B cell and T cell. And we know that B cell and T cell, both of them are called lymphocytes. Lymphocytes. Those lymphocytes, in order to work, they must know what antigen I'll be fighting. They must know what antigen I'll be fighting. That's why the macrophage will act like an APC. And this is the second function of macrophage. So the first function was phagocytosis. The second function was APC. When it introduced the pathogen on its surface, you must have a second transitional phase where B cell and T cell get to know the antigen. They must be activated by something called T helper cell. This T helper cell will introduce the antigen for both T cell and B cell. But in order for the T helper to introduce it, you must know that T helper have a binding site that can fit to the antigen. So if you can draw T helper like this and has a binding site, we can call this, generally speaking, as T helper. So this is the binding site of the antigen. So T helper can easily bind to this antigen by this binding site. Let's add another slide. Okay. So now we are talking about the third function of the T helper, of the macrophage, sorry. Now this is the T helper, I said that. With the binding site together, we consider them as inactive T helper cell. And this is the macrophage. This is a macrophage. And the macrophage, during the last process, it managed to take the antigen and present it on its surface. Antigen. So this macrophage right now is known as an APC, an antigen presenting cell, an antigen presenting cell. When this antigen binds to the inactive T helper, it looks like this later on. It will look like this. The T helper will manage to bind to the antigen, which is located on the surface of the macrophage. Now they bind. So far, we call this once again as an inactive T helper. This will take us to the third process or the third function of the macrophage. Macrophage now can release a chemical called interleukin. Write it in a proper way. Enter. Interleukin 1. This interleukin 1, when released from the macrophage itself, now the T helper can be dissociated from the macrophage along with the antigen, so that it would look like this. This is the T helper with its binding site, but this time it has the antigen binds to it. It has the antigen, and this part is called the T helper. But now this is called an activated T helper. Why is it activated? Because now it's released from the macrophage. And if you looked at the macrophage, they will turn like this. This is the macrophage without the antigen. Macrophage. Now it's not without the antigen. Why? Because the antigen is now carried with the T helper. If you have T helper like this, now the T helper can go and activate both cells, B cell and T cell. Now it can activate them. So all of this, but here we can call this the activation process. Number two, engine presenting cell function. Number three, activation of T helper. Activation of the T helper. Okay. And this is called the transitional phase. The phase between what? Between the second line of defense and the third line of defense. You must go and pass by the transitional phase. Thank you.